Hello, and welcome to AMiNDR, the podcast in which we sort and summarize the latest Alzheimer's disease research literature. So today, I am your host. My name is Naila. You may hear the occasional contribution from my cat, and I'll be going through papers published in August 2020 on preclinical testing. These are papers done in animal models that seek to target and treat different aspects of Alzheimer's disease. I hope you enjoy.
Welcome to AMiNDR, a podcast where we summarize the latest publications on Alzheimer's disease for you, so you can spend more time doing awesome research. For every month, you'll find a series of episodes by theme, and each comes with a bibliography. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast useful and accessible.
In today's episode, I'll be going through 11 papers that were published in August 2020 on preclinical testing. So these are peer-reviewed research papers that appeared on PubMed in August. And that's a lot of P's to be saying. I hope my microphone isn't picking them up too much. We have one paper on targeting the cholinergic system. Then we'll go on to targeting synaptic function, tau phosphorylation, and toxicity, amyloid-beta mediated toxicity and accumulation, and then we have two papers that don't really fit into any target system. So first up, we have one paper on targeting acetylcholinesterase / the cholinergic system. The cholinergic system is one of many hot topics of research for treatment, as the basal forebrain cholinergic circuitry is affected early on in Alzheimer's disease. Positive allosteric modulators for M1 muscarinic acetylcholine receptors are being investigated for their therapeutic potential, and you've likely heard us mention them in previous episodes. So they're often referred to as PAMs. So these PAMs have been reported to enhance cognition in multiple animal models of AD and could also improve the disruption of sleep-wake cycles or states that occur in AD, as these result from changes in the cholinergic system. So that's what this paper tests. Paper number 1 is entitled Modulation of Arousal and Sleep-Wake Architecture by M1 PAM. Then there's a long number, which I'll spare you, across young and aged rodents and non-human primates. The first author of this paper is Gold, the last author is Jones, and it was published in Neuropsychopharmacology. And this is a good place to note that if you want to find any of the papers we list here, you can sign up to receive our bibliographies, which have the full details of those papers. This study recorded EEG in mice, rats, and non-human primates to examine the effects of the selective M1 positive allosteric modulator, or PAM, on sleep-wake architecture and arousal. And if you haven't heard of EEG, that's electroencephalograms. They compared the new PAM drug, which I'm going to refer to as PAM-V. It has a very long name that I don't want to repeat. So they compared this PAM-V drug to 3 other drugs: the acetylcholinesterase inhibitor donepezil, an M1/M4 agonist, and another M1 PAM. The PAM-V produced dose-related increases in high-frequency gamma power, which is a correlate of arousal and cognition enhancement. This was true in young wild-type mice, rats, and non-human primates, but not in mice lacking the M1 acetylcholine receptor, which confirms the site of action. There were no cholinergic-mediated adverse effects, in contrast to the other drugs that were tested. In aged wild-type mice, PAM-V was also able to attenuate age-related decreases in REM sleep duration. This paper lends support to the idea that developing selective M1 PAMs could help in treating age and dementia-related sleep disturbances. That's it for targeting the cholinergic system in August 2020. I said that we would next cover the glutamatergic system, but I actually don't have any papers in this category, so let's move on to targeting synaptic function. We have 2 papers in this category for this month. The first targets the signaling pathways of the tropomyosin-related kinase B receptor, which is important in determining structure and function of neurons, including synaptic plasticity. And you might have heard of this receptor as TrkB. rather than tropomyosin-related kinase B. It's been shown that TrkB receptors are reduced in patients with AD, so activating the TrkB signaling pathways is a potential treatment strategy. So paper number 2 of the episode is entitled LMDS1, a potential TrkB receptor agonist provides a safe and neurotrophic effect for early phase Alzheimer's disease. This paper was published in Psychopharmacology by first author Fan and last author Xie Li. The authors wanted to identify potential TrkB agonists that could activate neuroprotective signaling and alleviate AD pathology in a mouse model. They started by testing 12 TrkB agonists in the cultured hippocampal neurons from mice, identifying 2 compounds that were particularly effective in restoring the dendritic arborization, or branching, of these neurons. These 2 compounds, termed 7,8-DHF and LMDS1, were then tested in amyloid-beta injected mice, and LMDS1 came out the winner. It improved short-term cognitive functioning and the pathology in these mice. The authors go on to identify several of the TrkB-dependent downstream effects of LMDS1 treatment, which includes upregulation of CREB and BDNF, and downregulation of the GSK3 beta active form and tau phosphorylation. So these are promising results for the potential of LMDS1 to treat some AD pathology, at least in mice. Moving on to paper number 3. This one is primarily focused on reducing A-beta and tau pathology, but it targets neuronal function in order to do so. The authors take a chemogenetic approach in which they manipulate the excitability of neurons, and this sort of crosses over to our non-pharmacological treatment episode. Some papers in there are also looking at stem cell and gene therapy, and we have a paper in there that's also on neuromodulation. Anyways, this one was published in PLOS Biology by first author Rodriguez, last author Husseini, and the title is Chemogenetic Attenuation of Neuronal Activity in the Entorhinal Cortex Reduces A-beta and tau Pathology in the Hippocampus. The authors tested whether A-beta associated hyperexcitability could influence tau pathology and propagation. To do so, they used a novel transgenic mouse line that models the impact of human APP and amyloid-beta accumulation on tau pathology in the entorhinal cortex hippocampal network. They found that human APP overexpression aggravated tau aggregation in the entorhinal cortex and accelerated its spread to the hippocampus. The human APP and A-beta caused neuronal hyperactivity in the entorhinal cortex, and when the authors used chronic chemogenic attenuation to reduce this hyperactivity, they also found that it reduced human APP and A-beta accumulation and reduced pathological tau spread. So overall, these findings support the idea that A-beta associated hyperactivity accelerates the progression of pathological tau along vulnerable neuronal circuits, and that chronic neuromodulatory approaches could improve AD pathology in vivo. Let's move right along to our third section today, which is targeting tau phosphorylation and the tau protein more generally. As you may already know, one of the key underlying disease mechanisms of AD is abnormally increased phosphorylation of the tau protein, which leads to tau pathology such as the formation of neurofibrillary tangles. This makes tau and its phosphorylation an interesting therapeutic target, as preventing this hyperphosphorylation could potentially prevent AD altogether, or at least mitigate some symptoms. So there are just 2 papers on targeting tau this month. Both papers use an antibody approach, which are also being explored for targeting A-beta, but you'll see they do so in quite different manners. So the title of paper number 4 is Contribution of Endogenous Antibodies to Learning Deficits and Astrocytosis in Human P31S Mutant Tau Transgenic Mice. It was published in Scientific Reports by first author Van der Hoeven and last author Ittner. The authors test two different approaches to depleting the B cell lineage and hence antibody formation in human P31S mutant tau transgenic mice. So these are the tau 58-2 mice, which I'll refer to as mutant tau mice. The first approach was crossing these mice with another transgenic mouse line, namely B cell deficient LGHM knockout mice. The second approach was treating the mutant tau mice with anti-CD20 antibodies that target B cell precursors. Both approaches led to B cell depletion, which significantly reduced astrocytosis in the mutant tau mice. Completely eliminating B cells using the transgenic mouse approach did also lead to some spatial learning deficits, but at the same time it improved motor performance. This was also associated with changes in brain region-specific tau solubility. Overall, this work indicated that the presence of antibodies throughout life contributes to astrocytosis in mice carrying a tau mutation, and while it limits learning deficits, other deficits and neuropathological changes seem to be independent of the presence of B cells and resulting antibodies. Paper number 5 was published by first author Vitale and last author D'Abramo. The title is Intramuscular Injection of Vectorized scFv-MC1 Reduces Pathological Tau in 2 different tau transgenic models. This was published in Acta Neuropathological Communications. And before I launch into this summary, just a note that this is an extension from the group's previous work. I guess most research papers are extensions on the lab's work, but what I want to say here is that the authors previously generated a vectorized single-chain variable fragment. This is what they abbreviate to scFv, which was derived from the conformation-dependent anti-tau antibody MC1. The resulting compound, if you can call it that, prevented tau pathology in adult mutant tau mice when injected intracranially, and in this paper they want to test whether it also does so when injected intramuscularly. So here the authors show that a one-time intramuscular injection of their vectorized scFv led to a long-lasting peripheral source of anti-tau scFv-MC1. I'm just going to refer to this as the anti-tau compound, so I don't have to say all those letters again. And this significantly reduced insoluble and soluble tau species in the brain. This was true in a prevention paradigm and in 2 different tau transgenic models, namely the JNPL3 and the P31S mice. The results indicated that the anti-tau compound was internalized by microglia without producing any overt inflammation. So as a refinement of this group's previous findings, this study demonstrates the efficacy of delivering the vectorized scFv through muscular tissue in order to target tau, and suggests that it could help treat AD and other tauopathies. And that brings us to the halfway point of this episode, so before we move on to amyloid-beta mediated toxicity, let's take a break.
Hey listeners! I'm here to let you know AMiNDR is recruiting. If you're interested in joining us, shoot us an email at amindrapodcast@gmail.com. You can also find us on Facebook, Twitter, and Instagram. Enjoy the rest of the episode.
Alright, and I'm back to tell you about 4 papers that target amyloid-beta mediated toxicity and production. So the other main AD pathology, as you likely know, is A-beta aggregation and the formation of plaques. And it's thought that A-beta mediated toxicity is a primary cause of AD. The next 4 papers take various approaches in targeting A-beta and trying to stop or reverse its pathological accumulation. So paper number 6 looks at stabilizing a transport protein called transthyretin, or TTR, into its tetrameric form. So TTR can bind to A-beta and reduce its toxicity, and this interaction is enhanced in the tetrameric form. This was published in the Journal of Alzheimer's Disease by first author Rake and last author Lopp. The title is Oral Treatment with Iododiflunisal Delays Hippocampal Amyloid-beta Formation in a Transgenic Mouse Model of Alzheimer's Disease, a Longitudinal In Vivo Molecular Imaging Study. Sorry for stumbling over that word. It gets trickier to say words after you've been recording for a while, especially pharmacological ones. The authors of paper number 6 tested the therapeutic efficacy of 2 TTR stabilizers, the first being iododiflunisal, which I'm going to abbreviate to IOD, and the second being tolcapone. And you can check the difference between the two in the abstract of this paper. They did this by using PET imaging in female APP/PS1 transgenic AD mice who received oral treatment at 5 months of age with either one of the TTRs or were not treated at all. The treatment efficacy was assessed by changes in longitudinal A-beta deposition in the hippocampus and the cortex, which was measured using the PET tracer 18F-fluorbetabine. The authors found that non-treated animals showed a progressive increase in cortical and hippocampal A-beta deposition between 5 and 11 months, which stabilized afterwards. In contrast, the PET tracer uptake remained constant between 5 and 11 months in mice that received IOD, but then increased significantly at 14 months. In tolcapone-treated mice, A-beta deposition did increase steadily, but at a much lower rate than in the non-treated group. These in vivo findings were also confirmed by immunohistochemistry at 14 months of age, and they suggest that these TTR stabilizers could reduce or slow amyloid-beta deposition in certain brain regions. Paper number 7 is on emodin, which, according to Wikipedia, is a chemical that can be isolated from rhubarb, buckthorn, and Japanese knotweed. Apparently, it's already shown potential in reducing the levels of beta amyloid, tau aggregation, oxidative stress, and the inflammatory response, so the authors wanted to test if it also affects A-beta accumulation. This was published in the Journal of Neurochemistry by first author Wang and last author Li, and the title is Emodin inhibits A-beta 42 aggregation and improves cognitive deficits in Alzheimer's disease transgenic mice. The authors studied whether emodin affected A-beta aggregation using a range of biochemical, biophysical, and cell-based approaches. Their findings suggest that emodin blocks A-beta 42 fibrillogenesis and A-beta-induced cytotoxicity and has greater effects than curcumin, which also often comes up in our drug development episodes. The authors also provide details on the interaction between emodin and A-beta, which you can find in the abstract in the paper. When they fed a high dose of emodin to 8-month-old transgenic AD mice for a period of 2 months, they found that it improved cognitive impairment by 60 to 70% and also drastically reduced A-beta deposition. So this sounds very promising for emodin. Okay, let's switch gears with paper number 8, which is on metals, such as copper and zinc, and these could worsen A-beta toxicity, so targeting them is the strategy of this next paper. It was published in Behavioral Pharmacology by first author DeFalco, last author Ray. The title is The Aryl Hydrazone INHHQ Prevents Memory Impairment Induced by Alzheimer's-Linked Amyloid-Beta Oligomers in Mice. Metal protein attenuating compounds have been proposed for treating Alzheimer's disease as they reduce abnormal interactions between metal ions with A-beta and could therefore inhibit A-beta pathology. In this paper, the authors investigate whether the isoniazid-derived aryl hydrazone, which is INHHQ, can protect against A-beta oligomer-induced memory impairment in mice. And sure enough, a single dose of INHHQ at 1 mg per kilogram administered systemically prevented both short and long-term memory impairments. The authors also conducted in vitro studies, finding that INHHQ prevents the production of reactive oxygen species that normally result from the interaction between copper and amyloid beta. The molecular mechanism remains to be determined, but these findings suggest that aryl hydrazones could indeed be neuroprotective agents in treating AD. Paper number 9 is the last one in this section, and it's quite technical. It's regarding the kinetics of A-beta and how they could be modulated by treating rats with calcitrol, which is the active vitamin D receptor ligand. This paper was published in Drug Metabolism and Disposition by first author Peng, last author Pang, and the title is Human Beta Amyloid 40 Kinetics After Intravenous and Intracerebroventricular Injections and Calcitriol Treatment in Rats in Vivo. The authors examined how pre-treatment with calcitriol changes the kinetics of human A-beta 40 in rats following either intravenous or intracerebroventricular administration. The authors provide several technical details regarding their human A-beta 40 kinetic assays. I'm not specialized in these, so check out the abstract for more details. And they also explain how they modeled the resulting data. Overall, the modeling of human A-beta 40 data following either administration route revealed an initially faster plasma half-life, reflecting faster peripheral distribution but slower equilibration between plasma and brain/CSF. So that's referring to the amyloid-beta 40 distribution. Pre-treatment with calcitriol increased the expression of P-glycoprotein in the brain, and P-glycoprotein acts as an efflux pump, so it makes sense that this also led to enhanced efflux clearance of human A-beta 40 from the brain. So let's finish off with 2 papers in the other category. So these are ones that didn't fit into any of the target system categories. First up, we have paper number 10, which is on dexmedetomidine, or just Dex. And this is an anti-anxiety sedative and painkiller. It could help treat cognitive impairment, and the authors wanted to understand how. So this was published in Molecular Neurobiology by first author Sun, last author Lee, and the title is Dexmedetomidine provides protection against hippocampal neuron apoptosis and cognitive impairment in mice with Alzheimer's disease by mediating the MIR129 YAP1 JAG1 axis. The authors set out to determine whether the microRNA precursor MIR129 is involved in the protective effect of DEX, specifically in A-beta 142 injected mice. When they tested cognitive function and hippocampal neuron apoptosis in these mice, they found that administering DEX improved both. This was accompanied by an enhancement of MIR129 expression, and the authors elaborate on the molecular interactions and pathways responsible for the resulting therapeutic effect. So specifically, as was in the title, they conclude that MIR129 YAP1 JAG1 axis could be the mechanism underlying DEX's neuroprotective effect in AD mice. And lastly, we have paper number 11. This is on estradiol replacement therapy, which relates very well to the non-genetic risk factors episode I recorded earlier this month. So if you're interested in how certain hormones could be protective or risk factors for AD, check that one out too. So paper number 11 is entitled Estradiol Replacement at the Critical Period Protects Hippocampal Neural Stem Cells to Improve Cognition in APP-PS1 Mice. It was published by Quinn, first author, or Kin, and last author Shah, in Frontiers in Aging Neuroscience. The authors tested the effective period for estradiol treatment using a mouse model of AD so as to better understand if there is a critical window for estrogen replacement therapy in postmenopausal women. 4-month-old female transgenic APP-PS1 mice were ovariectomized. I'll refer to these as OVX mice. And then they were treated with estradiol for 2 months. This started at either the age of 4 months, this is the early period, 6 months, mid-period, or 8 months, late period. The authors found that compared to age-matched wild-type mice, APP-PS1 mice with intact ovaries showed a progressive increase in A-beta accumulation with age, which was paralleled by reduced survival of immature neurons. The estradiol depletion caused by ovariectomy elevated A-beta levels in APP-PS1 mice and caused some other pathological and cognitive changes. These adverse effects were only prevented when the OVX mice received estradiol treatment right away, so that's in the early period, but not in the mid or late period. There are several mechanistic details I've excluded, so I suggest that you check out the abstract and the paper for more information. And that rounds off our preclinical testing episode for today. As a quick reminder, if you want to follow up on any of these papers, you can check out our bibliographies. You can receive these by signing up to our mailing list, and you can check the episode notes for details on that. I hope that you found this episode useful and accessible, and we're always happy for feedback on how to improve and how to best cater to our audiences. So you can get in touch with us by email. You can also find us on Facebook, Twitter, and Instagram. And I'd like to thank everyone who has worked very hard in producing, this and other episodes. Sarah did most of the sorting of these papers, and I summarized and scripted them. And the music that you heard at the beginning of this episode, as well as in the break, and that you'll be hearing right now, is by Anusha Kamesh. She is a fellow podcast host, a good friend of mine, and I recommend that you check out her music at either SoundCloud under Anusha Kamesh or on YouTube under AK Music. And lastly, if you would like to join our team, if you're interested in sorting papers or summarizing or even podcast editing and hosting, please email us with your CV and a quick note about how you'd like to contribute. That's it for me. Until next time.